Uh, so today we have to start our new enzyme, and before going to the newer one, we just go through what we have done yet, and then we'll start. So we started with the so we started with the history, then different types of enzymes. We talked about nucleases, endonucleases, then specific type of endonucleases that restriction enzymes. We talked about their classification, their properties, their mode of action, what type of cut they produce, different important enzymes, and their recognition site, and how we name them, and their isomeric mechanism in them. Then we talked about ligases, how they act, how they function, what their requirement, what are their utility, mechanism of action. There, then we talked about the polymerases, DNA polymerase, as the same as it you do in case of your DNA replication. Then we talk about different types of the polymerases, their mode of action, what they require. A simple thing they require the polymerization they do on a pre-existing strand. That's why we're calling the semi-conservative DNA replication. So that thing is there in case of your DNA polymerases. And yes, one more thing was there that they need a primer. If primer is not there, then polymerase is not going to function. And different type of polymerases, DNA pol 1, pol 2, pol 3, these are the three polymerases that are present in your bacteria. So from there only we get these polymerases. And alpha, beta, gamma, delta are the polymerases that are found in the eukaryotic system. And then the fragmentation of your DNA polymerase, DNA pol 3 that we talked about, it had different, yes, here you can see different types of the kinase, that is polymerase or nuclease activity. So which domain have first type of activity? So we segregate treating the enzyme name as the subtilisin, and the who worked on it. The Kinase is the name of the scientist who was working on it. So on the name of the scientist we call the Kinase fragment, and this is the Kinase fragment. Then we talk about the thermostable polymerases. So thermostable is the one that you have to utilize for the PCR. Thermo means temperature, means they have the stability at high temperature. And a few examples of the thermostable polymerase that we call the Taq polymerase. We originate from the Thermus aquaticus. And we mentioned that it have one drawback, that it can't proofread. We need a proofreading activity also, because you don't want any wrong incorporation of your base. So for that, some enzymes are there which are having this activity. Example was the PFU. And other than the PFU, there are some more enzymes which are thermostable. And depending upon your requirement, you can get these enzymes also. But most commonly used thermostable enzymes are your Taq polymerase and the PFU. And this was the end of the slide that we have done yet. And before moving to the next, if any one of you have any query, please raise now. Any one of you have any query till what we have discussed? Am I audible? Am I well audible?
Yes, sir, you're audible.
Okay, so do any one of you have any query?
Sir, you know how we use the best used polymerase is Taq polymerase, right? So can it be replaced by any of the other thermostable polymerases?
Yes, you can replace with any of the thermostable polymerase that you want. But thing is that what your requirement is, what your utility is, for what purpose you want to use. That the only that you select, and also the cost. So the Taq polymerase is very cheap. So that's why for any of the type of experiment we are doing, general experiment, general PCR polymerase experiment. So we use the Taq polymerase. But some specific thing that when you want to clone a gene, so when the cloning means you need a gene sequence that is present in the cell or in the DNA, so that you don't want to have any error in the amplification. So for that you can't rely on the tech polymerase because you do not have the proofreading activity. So for that purpose we work on PFU. And also you can say, you also know that when you want to go for the sequencing, in DNA sequencing, which is going to tell you the what sequence of DNA is. So in that case, if your polymerase readout is wrong, then that is going to be affect your sequence, because you are going to end with the wrong sequence. So depending upon your utility, we choose our DNA polymerase. So most commonly, as I mentioned, two are the tag and PFU are being used. Where you need a proofreading, you use the PFU. Where you don't need the proofreading, you use the tag. Okay.
Okay, sir. Thank you.
Okay, so now we are moving to the next type of the polymerase. That's the name is the polymerase reverse transcriptase, or simply you call it the reverse transcriptase. So as we are talking about it briefly in our last lecture also. So as the term transcription is there, so transcription simply means the formation of DNA to RNA, that we call the transcription. And now you're seeing reverse transcription means forming DNA from the RNA is going to the reverse to the transcription. So this is a just a minute, just a moment, hold on please. Okay, sorry. Okay, so we were talking about the transcription. The transcription I mentioned from the DNA to RNA. So reverse process is called the reverse transcription. Means you are forming DNA over a RNA. So one thing is there that you know in the both the processes, they are occurring on a template. If the template is not there, they are not going to function. So means they are dependent on the template. The polymerization activity that you are referring, that is depending on the template. So in the former case, when we are talking about the DNA polymerases, your template is going to be DNA. So that's why you call it as a DNA dependent DNA polymerase, because it is polymerizing DNA. These reverse transcriptase also the form the DNA, but they need a RNA template. So we call them RNA dependent DNA polymerase. Or simply we don't use to call so long name. Just by convention, we are using term reverse transcriptase. Okay, but true name of them is the DNA dependent, sorry, RNA dependent DNA polymerase. Okay, so that is the thing. So as I mentioned, what are the requirement? So they need a template. So template here is going to be RNA, and as I mentioned that they are the DNA polymerase, so they can't synthesize a strand de novo. The same problem is here also in this DNA polymerase also, that if you are saying that if you are having a RNA but you don't have the primer, they are not going to function. So they also need a primer. But as there is only single strand, you don't need two primers because you need to amplify just only one strand, so only one primer is required. So we didn't much talk about the primer, so I'm not talking about which primer we are going to use, what are going to be the utility. So when we will talk about the primers, in that time I will ask this question. Okay, so here is also one primer is going to be required, and as we all know that the DNA polymerization occurs in the 5' to the 3' direction. So here is also going to be the same 5' to the 3' direction of the newly synthesized strand. So if the RNA, it is going to be the RNA that you are having. So replication or the polymerization is going to start from the 3' end of your RNA, not from the 5' end. It is from the 3' end of your RNA, and that is going to amplify into the DNA. So requirement wise, whatever you need in case of a DNA polymerase, all things you need here also. That is your template you required. You required primer. Your DNTPs, nucleotide triphosphate you need. So and nucleotide triphosphate they are of deoxy in nature because oxy are going to be a ribose. So ribose you don't need, you are making DNA. So you need DNTPs. So whatever the requirement was there, here also all are the common. So as you can see, the smaller one alphabetically we are representing as a ribose nucleotides, RNTPs, but here are the capital ones are of the DNTPs. So lower is the RNA, and when it is going to amplify, it is going to form a complete complementary DNA. So this is the things now coming to the source from where we are going to get it. So these are you are not going to get from the your bacteria. So they are the source of these are the viruses because they are the part of viral replication system. Now we all are talking about COVID, and you all might be aware about that. This COVID is a RNA virus. So when it is an RNA virus, so it is going when it enter inside you, this RNA is going to change itself into the DNA. So the job of conversion of RNA to DNA is done by the enzyme that is present inside the virus. The virus are going to have a gene that is required for their replication, and those are RNA virus. They are only going to have it. For them, we call them the reverse transcriptase. So the source is a virus in this case. Earlier enzyme what we have talked about, like your DNA polymerase, your restriction enzyme, like this is all were in the bacterial origin, but this enzyme is going to be virus origin or viral origin. Okay. Okay, so the ability enzyme, as I mentioned, complementary. So here you are just DNA is going to be complementary to RNA. So that's why we call it as a cDNA or the complementary DNA because this DNA is complementary to RNA. So that's why we're using term complementary. All DNA, whatever your DNA polymerase is going to synthesize or RNA polymerase are going to synthesize, all are doing a complementary one. Whatever the strand you are going to get is a complementary. But here, in case of the RNA polymerase, you are going to have a single stranded DNA. So that's why we are calling it a complementary strand. This is a strand. Okay, it's single stranded one. So this is that's why we call it as a cDNA or the complementary DNA. Okay, so this term complementary, we are going to use it further when we are going to talk about much more about your uh, recombinant DNA technology, how we are going to utilize it. Okay, so this was a bit about your reverse transcriptase. So next enzyme is going to be your topoisomerase. So before moving to topoisomerase, if any one of you have any doubt, any query regarding the reverse transcriptase, please ask. Any query regarding reverse transcriptase?
No.
Okay, that's good. So the property wise, whatever the DNA polymerase protein is going to have, your reverse transcriptase is also going to have similar property. Only the template, but they are going to amplify, that is different. Okay. So as I was talking about COVID also, so in case of all of you are talking about the RT-PCR. So that RT-PCR came from this reverse transcriptase. R stand for reverse, T stand for transcriptase, ECR, reverse transcriptase for polymerase chain reaction. So you are going to utilize this reverse transcriptase enzyme in your COVID diagnosis. Okay, so this is simple an application where you can utilize, and two application. One is wherever you want to make a complementary strand from RNA to DNA, we are going to utilize reverse transcriptase. Once the DNA is going to be formed, next step, double-stranded form if you want to make, or if you want to further amplify it. So for that, you are going to need DNA polymerase. Reverse transcriptase is not going to work further. Reverse transcriptase is not going to form a double-stranded DNA. Reverse transcriptase is only going to form a single-stranded DNA. Okay. Okay, so next enzyme is the Topo Isomerase. So as you can see here, one term is the topo is there. So what do you mean by topo? Or also called the topology. What is topo? What is topology?
That's regarding anything regarding the surface. You call
Yes.
it's
It is regarding the surface, what the surface architecture is. Like some of us are fat, some of us are going to be lean also. Like my hand is like this, and now the topology is like this. Now this topology is going to be changed. So nothing is, my hand is going to be remain same. Just surface architecture is going to be changed. Like earlier it is expanded, now it in the form of fist. It is band one or folded one or compacted one. So this is the main difference. This is the topology, the surface architecture of my hand, how it is there. So in the one form it is like this, and another like this. So when I'm talking about this, you all know about the isomerism also. So what is the isomerism? What is isomerism? Ishita, Ishita, Ishita is sleeping, I think. Okay, Ayushi, Ayushi. Yes, Yuvraj, you want to say something? Yes, Yuvraj.
Yes. Yes, sir. The molecules which have the same number but different physical and chemical properties.
You must speak a bit loudly. Your is not.
I'm audible, sir?
You are audible, but not voice is not clear.
Sir, I'm trying to say that the molecules which are having same number but different physical and chemical properties.
Okay, so he's saying something same number of the something and something. I'm getting that what he's saying might be some network issue or a connection issue. Okay, so who other than Yuvraj is going to tell about the isomerism? Yes, Manaswini?
Sir, so when two substances they have the same chemical formula, but they have a different structure, as in the atoms are arranged differently, then that phenomenon is called isomerism, and the two substances will be isomers of each other.
Yes, so simply we can say that the same thing, if they are having two different forms, we can call them the isomer. Like stereoisomers are there in the space, how they are being arranged, we call them stereoisomer, or the different types of the isomers are there. So here also the isomer is there on the basis of the topology. So like my hand is there, so this is, and my hand is going to be the same, like this or this. But there are two different forms. These two different forms are the topological forms. So if two things are differing in their topology, then you are going to call it as a topoisomer. Okay, so here are enzymes. This enzyme we are going to talk about is name of the enzyme is isomerase. So means the enzyme is going to change the isomeric forms. So it is going to change the isomers. So isomer, what type of isomer it is going to change? It is going to change the isomer which is going to be different in their topology, surface geometry. Simply say, you can say if it is changing the surface chemistry, then you call they are the topo isomers. So the job of this enzyme is going to change the topology. Okay, so those enzymes we call the isomers. So now what type of the topology is there in case of our DNA? In case of our DNA, you might have heard about the term supercoiling. So DNA is not supercoiled. So here is a slide is going to explain you what type of the thing, what is a supercoiling, and this supercoiling is just when the DNA is circular and this DNA is a double helical, and when it is going to be circular also, then it further coils up. So further coiling is called the supercoil. And whatever DNA we are going to have, all of us, our DNA is going to be supercoiled, and most of us DNA is negatively supercoiled. Okay, so whatever DNA we are all are having, our DNA is also supercoiled. It's not just a property with the circular DNA that they are supercoiled. Our DNA, that the your chromosomes, they are linear one, but still supercoiling is there. And as I mentioned, this supercoiling is a property of a circular one. In our case also we have a local circularization or local circular form. And this local circular form is not just like a circular DNA, but this local circular form forms because of your association of your DNA with the proteins that make a local circular form. So that local circular form is actually the supercoiling. So supercoiling, you can see that in the just a moment. So you can see that this is a linear one, this is a circular one, and this circular one, if you are further coiling it, like simply if you take anything like my this one is there. So this is a linear one. If you turn it like this, so then you're further coiling. So this type of things we call it is a super coiling. Now the super coiling can be of two way. If you're turning like this, or if you're turning like this, which handed you are going to change. If you are going to right-handed or left-handed. So this way you can change it whether it is a negative supercoil or a positive supercoil. So you can see that the positive supercoil is going to be when your chi is from the left to right, and if it's from the right to left, then it is going to be negative supercoil. So this is the main difference between the negative and the positive supercoil. Simply you can say if DNA is crossing over it, then it is a negative supercoil. If DNA is coming behind, then we call it as a positive supercoil. So this is a simple just a terminology how it is being supercoiled. And one more thing here is that is called linking number. Linking number is a number which is going to describe you how much supercoiling is. But the negative supercoiling is a positive supercoiling. So linking number tells you the number of supercoils. So as I mentioned that these coils supercoiling are going to form when you are twisting them. Okay? Like this, if you are going to twist it, then one supercoil is going to be formed. But when you are twisting it, one more property came into the picture that is called the width. So width is going to be like it's like when we are coiling it, then it is further going to change itself. So this is called that is called the width. Okay, so supercoil crossing the axis, how much axis is going to be closed. So this twisting number and the writhing number, they both correspond to the your linking number. So linking number is going to be sum of twisting number and the writhing number. So this is your linear one, so nothing. So now you give the one twist. So as you give the twist, this is the negative twist, so negative one, positive twist, positive one. So this negative positive is depending on in which direction you are going to give. So here index one, so now to get left with this twist, it is going to change its shape. So this you call the width. Now width is 1. So negative 1 or the positive 1. So both twist and width are going to give you the linking, your linking number or your final super coiling. Okay. So this is a few brief description about the supercoiling. Now this supercoiling is a change in the topology surface. So this is a circular one. It is well appeared like circle, but it is like appearing like your 8 type of appearance. So this is a change in the topology. So the enzyme which changes the topology we call the Topo Isomerase. So enzyme which is going to change the topology of DNA, those enzyme we call the Topo Isomerase. So what they can do? They can make a non-supercoiled to supercoiled, negative supercoiled to positive supercoiled, or positive supercoiled to the negative supercoiled, or change the supercoiling linking number. If the linking number is earlier 3, if linking number changes to 2, that also a job of your Topo Isomerases. So how they act? So simply you can say that in the former one. So in this case, it is a non-supercoiled. It's supercoiling is 0. So in this case, one positive supercoil is there, one negative supercoil is there. So total linking number is going to be 0. So if you want to change the linking number, so it means one negative, one positive is there. So if you want to incorporate one negative coil, this positive coil to change to the negative coil. So for that, what you can do, you can break this phosphodiester bond between them and change. Like in this case, if you see, in this case, it is coming from. So it is moving. So it is moving from left to left and right. it is moving. This strand is moving from left to right, and here the strand is moving from right to left. The strand is moving from left to right. We are going to call it as a positive supercoil. If it is moving from left to right, we are going to call it as a negative supercoil. Okay, so this is positive, this is negative. So you want to change this positive to the negative. So what you can do? You have to break it so that lower strand is going to come forward. So now this strand is also moving from left to right. So right to left. This is also moving right to left. So both are the negative supercoil. One negative. So total is going to be -2. Okay. So this job, this breaking the phosphodiester bond and again joining. Before the joining, it have to move this strand which was above, and now it comes below. Okay, so what was below, it moves up, or it can do also. While it is not moving up, it is opening it, so it can be like this also. So in this case, linking number is going to be 1, 1 negative 1. So this job is done by your uh, Topo Isomerase. Okay, so there are two types of the Topoisomerase, type 1 and type 2. They depend upon what type of the cut they produce, what type of the job they are doing. If they are breaking only one strand, then you call them a type 1, and if they produce cut in the both the strand, then you are going to call them the type 2. Okay, so these are two different category of topoisomerase, type 1 and the type 2. So job, they can introduce a negative supercoil, or they can decrease. Both the type they can do, whether you are introducing a introducing a supercoil or you are decreasing the supercoil. So positive and negative supercoil, both your topoisomerase they can do. So now what role they get going to play, how they are going to be useful in your recombinant DNA technology. So while you are performing cloning experiment, so cloning experiment you utilize plasmids. So plasmids, they are going to be a supercoiled one. So these things, and one more thing is that this topoisomerases, they are going to have ligase activity also. So they promote, they make your cloning much better, so that you can do utilizing the topoisomerases. So this is about the topoisomerase, their utility, and when you want to reduce the supercoiling in your DNA, or they are going to help you in the cloning. So the cloning where you apply the topoisomerase is called the topo cloning. Okay. So now, if anyone of you would like to ask anything about the topoisomerase, please tell me.
Sir, can you please explain it once again, the positive and the negative?
Sorry, Ishita, your voice is not much audible to me.
Sir, the positive and the negative part, the topoisomerase, and can you please explain it once again?
What should I explain? You have to tell me specific thing what you didn't get.
Is right or left? Can you please explain
Ishita, do
that
one
part?
thing. Type in the chat box. Your voice is
Okay, sir.
not clear to me. Type in the chat box. What do you want to for me to explain again? Okay, so meanwhile Ishita is typing. Anyone other than Ishita want to ask anything? Okay, so topoisomerase cloning you want to know. So topoisomerase cloning in detail we will study later also. But the topoisomerase cloning is one, as I mentioned you, that in the topoisomerase cloning, what is going to occur? The ends of your vector, they are linked with a topoisomerase enzyme. Okay, so like a vector that we have talked about in your laboratory, like this is a circular one, and when we open it, so it's become linear one. So like this is a closed one. So if you open it, yes. So this is a vector. If you open it, so it is going to be like this. So like this, now it is open. So now to this opened end, you have added to both the ends. You have added your topoisomerase. So topoisomerase, what is going? It is going to help in the cloning. It will increase the efficiency of DNA ligase. It will assist the DNA ligase because it have the property of relaxation. Like if there is any supercoiling in your DNA, it is going to relax that, and that after relaxing, it will your ligation is going to be efficient. Okay, so it is going to help in the ligation. So Yukta also want to ask how do we calculate RIT? So RIT is simply as you can see the image. So in this image, this is the one. Like it is like this. So as it twisted like this, this is the one RIT. So how many times it is crossing the axis? We call the one RIT. So here you can see one. Just I will take back. So here width is going to be 1, 2, 3. Width is 3 and it is going to go from the right to left. So when it is going right to left, it is going to be -1, -1, -1. Okay, and the linking number is a total supercoil. So width plus twist is going to give you the your total supercoil. And next in the last slide you explained on the positive and negative topoisomerase. No, I didn't call the positive negative topoisomerase. I call the positive and negative supercoiling. I talked about the type 1 and type 2. Type 1 which produce cut in the one strand, and type 2 which is producing cut in the both the strands. Okay, so I think this it explain the your questions. What you are asking? Okay, so Yukta is saying okay.
Sir,
And yes, Shakti.
sir, I have a question.
Yes, Shakti, while you are asking, please also turn on your camera so that it look like that I am talking to someone, not to screen.
Yes, sir. Am I visible?
Yes, now you are yes, you are visible.
Yes, sir. So, what is the need to change the topology?
See, need of the change of the topology for the relaxation. Like the need of change of the topology is during your transcription and your DNA replication. Like if your DNA is twisted like this, like simply if you say, if your DNA is twisted like this, and you want to replicate it, you want to unwind it, you want to make double stranded to the single stranded, it is not going to be formed, it's not going to happen. Okay, so for that, if it is like this, then you can open it easily. So in this form you can't.
Yes, sir.
Okay, so that is the why you need to topoisomerase. These topoisomerases are there. They are the main. Their function. They function during the both the DNA replication and the transcription.
Okay, sir. Thank you.
Okay. So these are the enzymes that are for your natural purpose, but we are also utilizing these enzymes for our benefit that we are while we are using in the recombinant DNA technology. Okay.
Sir.
Yes.
So, suppose we have a positive sense DNA, and then we are turning it into negative sense. Like, does it make any difference?
See, most of DNA that we are going to have, it is a negative supercoil.
Hmm.
So for it is not going to make any thing that for us if we change from the negative to positive supercoil. But for our study, you want to check whether if you are dealing with positive supercoil, then how it is going to change your function. So that it is, but as per function, negative and positive supercoiling will not have much role. Their main role is of profiling is just decreasing the space. So here you can see if the DNA is like this circular, it is occupying more space, wherever it is occupying less space. So the purpose of supercoiling is bringing DNA into a lower surface area.
Okay, sir.
Or lower volume, you can say. Surface area not going to change, lower volume. Okay, so anyone, any other? Okay, so if no further questions, then we can move further. Okay, so next type is then we're going to talk about next type of enzyme that we call the DNA modifying enzyme. So this is the last class of our enzyme that we are going to talk about. They are going to modify your DNA. So when we say the modifying our DNA, it means we are going to add some group or remove some group from your DNA. So as you might all aware about the structure of your DNA also. You'd have a phosphate group. You'd have a OH groups at different locations. More specifically, 3' OH is important for us. And it has the nitrogenous base, and nitrogenous base are going to be GC. They are describing them on the basis of what type of the functional group they are going to have. So now, if you want to change somewhere, what type of change you can do? You can remove the phosphate group. So if you remove the phosphate group, then whatever ligase you will add, it won't combine, it won't join two strands. And similarly, if you remove the 3' OH group, so nowhere replication is going to occur. The polymerization, DNA polymerase won't function, and DNA ligase is also not going to function. And some other type of modifications are also there, like your methyl group. If anyone of you remember the structure of uracil and the thymine, sorry, uracil and yes, thymine. So what is the difference between structure and uracil and thymine? If anyone of you remember? Yes. Can anybody tell me about any difference between the structure of thymine and the uracil? A pair with U also and A pair with the T also. In RNA it is U, in DNA it is T.
Sir, thymine has an extra methyl group.
Yes, it have an extra methyl group. So if you remove that methyl group, then it becomes the uracil. Okay. But the uracil, if you add the methyl group, then it becomes the thymine. So like that. So you can do that type of a thing. In case most of the modification in DNA, if you talk about, there is the methylation. Methylation of the cytosine. It's not the thymine and the uracil. Methylation of cytosine. Cytosine not having the methyl group, but you can add additional methyl group to cytosine. Cytosine means we are talking about the nitrogenous base. Here we are not talking about your sugar part. Okay. For the sugar part, we are saying that you can remove the phosphate group and you can remove the OH group. You can add the OH group or you can add the phosphate group. So these are the some modification that you can do in your DNA. So whatever the type of enzyme which are performing such type of action, we call the name. So one most important enzyme first is the name the alkaline phosphatase. So here the term alkaline is there and the phosphatase is there. So two types of the enzyme are there. When one is called the kinases, and second type of enzyme is called the phosphatase. So where you at the term use the kinase. So term kinase is being used for the utilization of ATP, and where the utilization of ATP is there, we are going to add the phosphate group. So kinase are those enzymes which are adding the phosphate group. Whereas to the reverse to that is the phosphatase. So phosphatase are those enzymes which are going to remove the phosphate group. Okay, so phosphate group is present at the 5'. If they remove the phosphate group, what result you are going to get? You are going to get an OH group because sugar is going to have only OH. So from the sugar, the 5', the phosphate was there, that is being removed, and now you are going to have just sugar. So the enzyme that is going to play this role is going to call the alkaline phosphatase. Okay, so remove the phosphate group that is present at the 5' terminus of your DNA molecule. So this is the job of the alkaline phosphatase. Okay, So the two 5' are there. So you can see this 5' and second 5'. So now because of this 5' you have removed. So now ligation is not going to occur at these two ends. Okay. So this is a job of your DNA modification enzyme, that they remove the phosphate groups and the phosphate groups will be changed with the hydroxyl group. And this hydroxyl group is, they are not introducing a hydroxyl group. Once they are breaking the phosphate bond, phosphodiester bond that we are calling, as you break that bond, that OH group is going to be regenerated. That was previously present at your sugar. Okay. Second one is the kinase. So kinase is a reverse to that. Means if you want to add, so you can add the kinases. So for that you need the ATP. So these kinases, they are going to add. So two enzymes that I mentioned, one is the alkaline phosphatase. Their job is to remove the phosphate group, and the kinase, they are going to add the phosphate group. So they are reverse to the alkaline phosphate. What they are going to do? They are going to add phosphate group at the 5' terminus. Okay. And the next type of next enzyme that is going to modification is called the terminal deoxytransferase. So name in this enzyme, if you read the name, one is the terminal. In your DNA, there are two terminus. One is the 5' terminus, and second is the 3' terminus. So we are going to talk about these terminus. At those terminus, what are we going to do? Deoxynucleotidal means you are going to transfer a deoxynucleotide. Means you are adding a nucleotide, and this nucleotide group you are transferring to the terminus. So the enzyme which transfer a nucleotide at the terminus is called the terminal deoxynucleotidal transferase. So you may ask that, sir, these are the addition job of the adding your nucleotide at the end is of DNA polymerases. So how this is different from the DNA polymerase? So DNA polymerase, they are polymerizing, they are adding the adding a nucleotide. So these enzymes are also adding a nucleotide. The difference between your DNA polymerase and the transferases are that DNA polymerase, they need a template. So they can amplify a DNA, they can extend a DNA over a template. They can't add additional nucleotide. But the additional nucleotide that you want to add, that job is done by the terminal deoxynucleotidal transferase. So now question came from where they came into the picture. Might be all of you are remembering the transcription. And the transcription in eukaryotes. So how the transcription in eukaryote differ from the prokaryote? How the transcription in eukaryote differ from the prokaryotes?
What,
How
sir?
the transcription differ? Yes, Pradyuman?
Sir, I'm asking what you're trying to say.
Pradyuman, your voice is not coming well. Please type. Other than Pradyuman, Sanjana, 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 Sanjana is absent, I think. So I will mark Sanjana absent whenever she came in the class at the end. Sanjana. Okay. So next I'm calling Shivani Mishra. Shivani Mishra.
Sir,
Yes,
she,
Yes, Shivani, tell me about the difference between the prokaryotic transcription and the eukaryotic.
yes, sir.
Difference between the prokaryotic and eukaryotic transcription.
Hello.
Shivani, difference between the prokaryotic and eukaryotic transcription, I'm asking. those who are saying that I am having a mic issue, I am going to mark them absent. They will correct this mic issue in next class. This is going to happen then only, otherwise this mic issue will sustain till end of the semester. Yes, Manaswini?
Sir, there are several differences between the two processes. So one difference is that the enzymes that are used, since we're talking about enzymes, they are a little different. So the enzymes in prokaryotic transcription, it's less complex as compared to what's there in eukaryotic.
That is different. That is a no. That is a different thing. That is not a difference. You can say they also have the enzyme. They also have enzymes. The different origin, definitely different enzymes are going to be there. What thing is
Right.
present in one but is not there in the others? They are going to be the difference. Yes, Ishita, want to say something?
Sir, am I audible?
Yes, Ishita, you're audible. You can also turn on your camera.
Okay, sir. So one difference is that in prokaryotics, the transcription takes place inside the cytoplasm, whereas in eukaryotics, it usually takes place in the nucleus.
प्रोकैरियोट में न्यूक्लियस होता ही नहीं तो वहाँ कैसे न्यूक्लियस में होगी?
No, sir. In eukaryotes, it takes place inside nucleus.
हाँ वही तो मैं कह रहा हूँ, प्रोकैरियोट में न्यूक्लियस होता ही नहीं है. अगर होता तो वहाँ भी न्यूक्लियस में होती.
Sir, in eukaryotes.
हाँ मैं कह रहा हूँ, प्रोकैरियोट में न्यूक्लियस होता ही नहीं है ना.
Yes, sir.
हाँ तो वो कोई
That's
डिफरेंस
why.
वाली बात नहीं हुई ना. उसमें है ही नहीं वो चीज.
Sir, can I answer? So, Shivam.
Yes, who is raised? Manas, this Shivani.
Hello.
Yes, bolo.
Sir, the difference is
Turn on your
that
camera
in
also. Yukta, turn on your camera.
sir, am I visible?
You will appear meanwhile. Keep on, keep saying.
Huh? Hello.
Continue.
Sir,
You
I'm
know-
saying that in prokaryotes there are exons, and in eukaryotes we have exon in introns too, and introns needs to be removed. So there is splicing of that. That's the difference.
Yes, one difference. Splicing is there. Any other? Yes, Manaswini.
Sir, so the eukaryotic transcription will need initiation factors, whereas prokaryotic won't.
All need. उसमें भी चाहिए होता है. So the major
Sorry.
difference is the post-transcriptional modification. In prokaryotes, you are neither adding cap, you are not adding a poly-A tail. Okay, so addition of a poly-A tail is the one that I am talking about. The main thing, the poly-A tail, and this poly-A tail is a new things. For this poly-A tail, your enzymes, that is the terminal transferase is the enzyme that lies there. That enzyme we are going to use here. Okay, so the terminal transferases that in your transcription, they do not require a template. Okay, so that's the thing that you want to add one or more DNTPs into three terminus, single stranded, double stranded molecules. So they are of depending on what type of strand they are going to act. The enzymes came, the single stranded transferase, double stranded transferase. So you can have. So as you can see, the 5' to 3'. So when you add, then it is going to add to the 3'. So one similarity between your DNA polymerases and the terminal transferases is that they were also adding at the 3 terminus. The replication is extended between 5' to the 3'. Here also the same 5' to the 3' is there. So the both we are both occurring in the both the strand. So then nucleotide is going to be added at the 3' of the both strand of your DNA. Okay, so Topoisomerase we already have talked about. So they produce the nick and they relax it. So this is the one that going to occur. And I think the time is getting over. So I'm stopping at this point, and the last topic is going to be remain, that is your adapters and the linkers. Adding adapters and linker and what are the utility, what are these adapters are there. These will talk in our next session. So now we are at the end of session. So for the attendance, so just a moment. I'm opting beyond. Okay. So when I call your name, so please turn on your camera. So Demetrius.
Present, sir.
Tridev.
Present, sir.
Maniswini.
Present, sir.
Adish.
Present, sir.
Yuvraj.
Present, sir.
Nehal.
Sir, MSc is hanging.
युवराज हो गया आपका. नेहल. 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 नेहल एब्सेंट.
Sir, sir, MSc is hanging.
क्या कह रहे हैं युवराज?
She suggested that her MSc is hanging.
आपकी आवाज साफ नहीं आ रही, आप टाइप कर दीजिए युवराज.
सर नेहल का MS टीम हैंग कर रहा है.
कोई बात नहीं है, एक दो बार एब्सेंट लगेगी तो ठीक हो जाएगा. ठीक है. ओके, सो अतोयबा. अतोयबा. अतोयबा एब्सेंट. Then Devanshi. Devanshi. Nandini.
Present, sir.
Kshitij.
Present, sir.
Janvi.
Present, sir.
Also turn on your camera. So Nandini, Kshitij, Janvi, Devanshi didn't turn on your camera. Shakti.
Present, sir.
Present, sir.
Rupal. Sahiba.
Present.
Sahiba.
Present, sir.
Ishita.
Present, sir.
Namrita.
Present, sir.
Tanvi.
Present, sir.
Yukta.
Present, sir.
Ayushi.
Present, sir.
And last is Vivek.
Present, sir.
Okay, so Athoiba is absent, the rest all are present. So stay safe, stay happy, good day.
Thank you, sir.
Thank you. Thank you, sir.
Thank you, sir.
Thank you, sir.
Welcome.